Hello, welcome everyone to the Arthritis National Research Foundation's Research Spotlight Series. I'm Kaylin Young, the Director of Corporate Affairs, and on behalf of our Board of Directors, our Scientific Advisory Board, and all of our staff, I would like to welcome you to the Spotlight on Sjögren's disease. We're excited to have so many of you join us from all around the world. But before we get started, we have a couple of logistical pieces of information we want to share. So first is this webinar is being recorded and we will have it available on our website and YouTube channel in the coming weeks. If you have any questions, please type them into the Q&A box. And if we aren't able to get to your question, please email info@curearthritis.org after the webinar with your questions. To get us started, we'd love to hear from you. So I'm going to launch a poll, and it's going to ask how you're coming to this webinar. So, are you living with Sjögren's disease? Are you a caregiver, a family member, a healthcare professional, a student, a researcher, or you're here for another reason? We'd love to just kind of see what our audience looks like today. Wow. So it looks like most attendees are patients, and that's a great place for us to begin. What we'll learn today will help us understand what's coming next in terms of diagnosis and treatment. Now, I am honored to introduce Dr. Koppes. Dr. Abigail Koppes leads the Advanced Biomaterials for Neuroengineering Laboratory, where her team is exploring how to repair nerve damage caused by conditions like Sjögren's disease. They're developing innovative materials and techniques to support nerve regeneration and better understanding how the nervous system is affected in Sjögren's. Her goal is to create new therapies that improve quality of life for people living with this condition and related disorders. I'd like to hand it over to Dr. Koppes.
All right, well, thank you so much for the invitation to speak here today from ANRF as well as the Sjögren's Foundation. I'm going to start off by talking a little bit about the disease for those of you who are not familiar with it. But Sjögren's disease is a systemic autoimmune disorder. It's often characterized, at least especially at the beginning, by things like dry eye and dry mouth. So that might be the first thing that you notice as a patient. This is often because autoantibodies and our immune cells will infiltrate into these glands, sort of cause this chronic inflammation and downstream tissue destruction. So this is sort of the first thing that you might notice. It's often also characterized as primary Sjögren's, which is when it can kind of occur alone, or secondary, which is when it presents with other autoimmune diseases, which I'll be talking about a bit later. In terms of how many people this affects, it's about 0.01% of the population, although we think this is underrepresented. It impacts more women than men, although it does impact men as well. And the peak age is around 40 to 60 years old. In terms of diagnosis, there's blood tests that can be done. So you might have heard of, SSA or Rho or SSB or LA if you're a patient. Biopsy of your salivary gland to see how much infiltration you have of these immune cells, dry eye test called the Schirmer test or staining, as well as measuring your saliva. One issue that we see a lot is that about 30 to 40% of patients are seronegative. Now, what that means is that these autoantibodies may not show up in your blood test, which often can lead to diagnoses such as, connective tissue disorder instead of a Sjögren's disease, but often the rheumatologists and other doctors will look at you holistically to see what your constellation of symptoms are. Over time, this disease can be slowly progressive, and over time, about 30 to 40% will have other gland symptoms. They'll have issues with aches in their joints. You can have lung damage as well as problems with your nerves, which is what I'll be talking mostly about today. In addition, you have an increased risk of lifetime lymphoma, which is higher than the general population, and that requires monitoring over time as well. In terms of treatment, that's kind of currently going on. We'll touch on that first. Usually when the symptoms are more mild, the first line of treatment is things like dry eye treatment with tissue or drops such as Restasis, which you might have heard of. There's ointments that you can put in when you're sleeping. either gels or a petroleum-based system. You can get punctal plugs put in your eyes to keep your tear production and your oil there, as well as things like saliva substitutes. Exovac, I think, is one that many people take in terms of helping you produce more saliva. As your disease progresses, some people might be getting hydroxychloroquine or NSAIDs. You can also have steroids or immunosuppressants. And as time can go on, if you have other disease presentations such as neuropathy or low antibodies, you may also get IVIG or SCIG, which is another immunotherapy. Right now, there are a number of really exciting clinical trials that are ongoing. I'm not going to go into the specifics of these today because I'm not involved in these studies, but we have a number of different monoclonal antibody treatments, peptide treatments, and right now there are some phase 3 studies that should be showing readouts later this year, which is really exciting. Now, importantly, part of the message is to really make sure you have a multidisciplinary care team. And I'll talk about my personal experience with that in a minute. But this could be, rheumatologist, ophthalmologist, dentistry, primary care. If you have dry mouth, seeing your dentist 3 times a year instead of 2 times a year can be helpful because we're not making saliva the way that the general public does. So the reason that I care about this is, I'm a scientist. I'm also a researcher. I grew up in Western Massachusetts. I work in Boston at Northeastern University. I'm 40 years old, and I started having symptoms of Sjogren's, which I'll talk about in a minute, when I was, oh, my slides are moving without me touching anything. So somebody else touching the slides right now? Okay, sorry, we're going to go back a minute here. So basically, with myself, can everyone else stop touching the slides? Thank you. So basically, for me, I was a really active person before. I was doing research all the time and kind of go, go, go. As I got COVID in 2020, basically what happened was I first started presenting with Sjogren's disease. I didn't know that's what it was at the time, but I had a Schirmer test of 0 millimeters, meaning I was not producing, any liquids from my eyes. I had corneal abrasions. I was choking on my food because my dry mouth was so bad, which was obviously really scary for those of you who can relate to that. I later found out that I also was presenting with post-viral autoimmune polyfiber neuropathy, meaning my autoantibodies, in addition to attacking my glands, were basically causing my nervous system to be attacked by my immune cells as well. So you can kind of think about this as like having MS, but for your periphery. And then I also found out that I had mast cell activation syndrome, which is when your immune system is sort of just turned up. It's all on blast, it's hyperactive, and this can cause a lot of these problems. So these diseases can often present sort of together, and we don't really understand why that is, or sort of the chicken and the egg of this situation. And so oftentimes you might have heard of this as termed neuro Sjogren's. So, as a researcher that studies these things, I took a look at my own journey and how I had to, set up this giant multidisciplinary care team. This is all the doctors that I have here that I'll talk, more about later if you'd like me to. But the number one thing that's helped me, which is not advice, but my personal journey, was I started having IVIG or immunotherapy for my small fiber neuropathy, which was progressive for over 2 years now, and I'm seeing results not only just in my nervous system, but also in my Sjogren's symptoms. So I have a lot of different treatments. I use resources from, ANRF and Cure Arthritis, as well as the Sjogren's Foundation, to, get information up to date, so that you can always be sort of checking out what's going on with the new research that's happening and the treatments that are coming out. But this was kind of my personal story, which led me to be really interested in studying how the nervous system can be impacted by Sjogren's disease. From my own experience, but knowing that not many people are studying this impact, what can we bring to the table to look at this in a different way? And so with Sjogren's autoimmunity, we know the nervous system can be involved, especially as the disease progresses, but we don't really understand why. We know there's inflammation. We know there's different proteins that are being made. We know your body has autoimmune disease. We also know that oftentimes you can have issues with like permeability in your gut that causes your epithelial barrier to break down, and then inflammation can become systemic. So our thought process and hypothesis overall is that there's something in the blood of Sjogren's patients that is then impacting this neuronal dysfunction, which can lead to things like brain fog and other issues in terms of systemic response, but also locally in terms of neuropathy. So our goal is really to build models so that we can better understand the disease, which will lead us to having targeted therapies to prevent or treat the disease instead of just sort of treating symptoms, which is often how treatments are going right now. The system that I'm most interested in studying for this reason is the enteric nervous system. Now, you might have never heard of this before, right? Most people are familiar with their brain or maybe the sensory nervous system, right? Because you're touching things, you're feeling things, or your motor system because you're using it to move your body through space. But the enteric nervous system is actually the nervous system that is in your gut and helps to connect your gut to your brain. So if you've ever had butterflies in your stomach, maybe you're giving a talk to a group of people like I am right now, and you're a little nervous, you might have that kind of feeling in your stomach. That's the nervous system that's sort of being active and telling you to pay attention. In Sjogren's disease, up to 90% of patients have some form from mild to severe gastrointestinal involvement. We also know that 80% of adults with CNS involvement or brain involvement in Sjogren's disease report cognitive abnormalities, including things like anxiety, depression, and cognitive dysfunction, as well as I mentioned, brain fog and things like that. So because there's this sort of, correlation but not causation identified, what we want to do is study how the cells are impacted in Sjogren's disease with different new models. Now, I'm just going to look at this slide really quickly, but basically what we're interested in is kind of shortening the time from the bench to the clinic. So as you all know, if you look at this slide here, we can see that bench studies, which is like cell culture studies, have a lot of different studies. And as you progress through time, over years, you go from bench to animal to human studies. This can take a lot of time and it takes a lot of money. And as it goes forward, you actually lose a number of candidates to this leaky pipeline. And so one of the things that we're interested in is using these systems called organ chips, which are shown on the right, which are these little handheld devices that we can basically model some of the human biology at an earlier stage instead of relying on bench and animal studies to look at this disease progression and biology, and also to aid and make the design space smaller for those studies like animal studies to happen faster. So the way that we're doing that is basically we build these microfluidic devices, which are tiny little sandwich-like devices. We use tape that's from NASA, and we use plastic that's like in a fish tank. And we basically stick them together, and we can make these little flow models that we can then put neurons in from our gut and add different things to study how the cells respond. So these allow us to build them cheaply. We can look through them, we can image them, we can take samples from them, we can make disease happen in them, and then we can study them over time instead of having to rely on other models that are more 2D. So this mimics the human physiology a little bit more closely than some of the traditional models that are used in pharma. Here is shown a picture in A in the center of what one of these little chips looks like, and next to it is shown what your neurons actually look like taken from a human gut. And so what we're really interested in doing is taking these cells and in collaboration with Sarah McCoy, who's a doctor, MD, PhD at the University of Wisconsin-Madison, she is giving us serum from patients that have Sjogren's, seronegative or severe cases. And what we can do is then use our platforms to study how the changes in the neuron activity happens. So looking at these little graphs on the right, you can see there's different dots, different lines, and basically that's a single neuron telling us it's active or not. And so over time, we can look at how they change and determine what is changing and which conditions of patient severity make our neurons behave differently. And we're really expecting that we're going to see changes in either their signaling to go increased or decreased, as well as alterations in how these signals fire, indicating that there is an impact on the nervous system itself. We're also looking at basically scaling this up. So as I mentioned, the pipeline from drug discovery from the day one to human use is really slow and really leaky. And so what we're able to do is take our organ-on-a-chip systems and make them fit into 12 plates on one single device. We then use fiber optic cables, just like you have getting your internet to stream this talk. And we can shine the light from the fiber optics at our cells, and we can see how they change over time. This is really exciting because we can get data from a number of cells shown on the right from single fiber optic cables. So you can see fiber 1 through fiber 7 in this graph, and each little peak is showing how a neuron is changing its cell signaling in response to a different cue. And so we can really now scale up this model and start to look at things and study questions like what components are specifically in the blood that is changing these neurons to fire, and how does progression of the disease actually work? So we're really excited right now to be doing these studies, and we hope to have a big readout at the beginning of next year, hopefully January, which we'll be reporting and we're happy to share with you all again in the future if you follow along. But we're really looking to identify both how the biology is happening and then use these platforms to screen therapeutics in a way that's faster than the traditional method, so we can get treatments out to patients in a quicker manner. So with that, I'd really like to thank the team that's working on this, as well as our collaborators, Dr. McCoy, Sjögren's Foundation, ANRF, and Northeastern University for your support. And I'm happy to take questions at the end with all the other people. So looking forward to seeing their talks too. Thank you very much.
Thank you so much, Dr. Koppes, for sharing your incredible story as both a patient and your impactful research. I'd like to remind all of our attendees to please put your questions in the Q&A box, and we will get to that at the end and make sure to answer them. We've already had a couple coming in, so we're really glad to see that. And now I have the pleasure of introducing Dr. Lieberman. Dr. Scott Lieberman grew up in New Jersey and first studied chemical engineering at Rutgers University before discovering his passion for both medicine and research. He went to earn his MD and PhD at the Albert Einstein College of Medicine, where he developed a strong interest in immunology and autoimmune disease. Today, Dr. Lieberman is a professor of pediatrics at the University of Iowa, where he combines patient care with research. As a physician scientist, he leads a research lab and has a particular focus on childhood Sjögren's, and co-leads an international work group dedicated to improving how this rare disease is diagnosed and treated in kids. In addition to his research and clinical work, Dr. Lieberman serves on the board of directors of the Sjögren's Foundation, and while his professional achievements are many, he is also known as an all-around swell guy who is deeply dedicated to both his patients, his science, and his community. Dr. Lieberman, I'll let you take it away.
Well, thank you very much. I don't know if I can follow that introduction, but a swell guy. I love it. So thank you very much for the opportunity to speak today. I appreciate the Arthritis National Research Foundation and the Sjögren's Foundation for having me talk about Sjögren's disease, which I think about all the time. So you kind of already heard a bit about me, but just so you can see the journey on a map. I grew up close to the Jersey Shore in Toms River, New Jersey. And then I went up to Rutgers College of Engineering, where I thought I would become a chemical engineer. But I found immunology research there and also found a love for pediatrics and clinical medicine. And so I went up to New York, and that's where in the Albert Einstein College of Medicine, I trained both studying medicine as well as really learning a passion for studying autoimmune diseases in the lab. And then in Philadelphia, when I trained at the Children's Hospital of Philadelphia, both in pediatrics and then in pediatric rheumatology, that's where I really was introduced to Sjögren's disease and just the world of things that we did not know about Sjögren's disease in children. It is often a disease that is diagnosed much later in life, as you heard already, but it turns out that it does occur in kids, and it often presents with much vaguer symptoms that I think if we are paying more attention, those of us who see children in rheumatology clinic, maybe we can capture this disease much earlier, but we need better tests. So then obviously, as you can follow my path, it's probably makes perfect sense why I would escape the Northeast and move to the Midwest. And so I've been hanging out here for the last 12 years, and I'll stay until they kick me out, I guess. So I thought I would take it back because really what I think about all the time is the immune system, right? And so I wanted to make sure we're all on the same page. So the immune system really is very complex, which I'm sure is a gross understatement, and you're all very aware of that. But we know the immune system has many different types of cells, and this is very important because they do different things. And in our lifetime, we will be exposed to many different germs or microbes. And it's important for the immune system, really, their main job is to attack these germs so that they can rid them from the body and then keep us healthy. But we know that in the case of autoimmunity, the immune system is essentially getting confused and attacking our own body. And then what we actually call the autoimmune disease that a person has is really based on which parts of the body are attacked. And there are some autoimmune diseases, as you've already heard, that can affect many different parts of the body and cause widespread inflammation. And this is why we call them systemic autoimmune diseases, because they can affect the whole system. So Sjögren's is a systemic autoimmune disease, and it can affect any part of the body, and it often affects multiple parts. And the timing of which parts get affected when really is something I don't know that we've been able to figure out how to predict, but it's something that those of us that treat children or adults with autoimmune diseases know you have to keep track of different symptoms and figure out if new parts are being affected. So one of the hallmarks of Sjögren's that really sets it apart from other conditions is the immune cells attacking these glands that make saliva and that make tears. And so in this next schematic, you'll see where these glands are. So the lacrimal glands, or the tear-producing glands, are present really in the orbit just above the eye, kind of behind the bone, and they produce a lot of the tears, and that helps to protect the eye so that you can prevent infections and you can maintain a healthy cornea and a healthy surface of the eye, which if you have Sjögren's and you have decrease in tears, then you can get damage, and that can result in pretty significant effects on being able to see. So the major salivary glands, or saliva producing glands, there really are three sets. There's the parotid gland, which you can see off to the right, and that's kind of the biggest one right underneath the tissue here in front of your ear, and just kind of hooking around the edge of the mandible, the jawbone. Then there's the submandibular gland, which is just in front of the parotid gland, but a little lower, kind of tucked under the jawbone, more in your neck. And then the sublingual gland, which is really below the floor of the mouth, kind of below the chin here. And these glands really are the major salivary glands, but it turns out there are actually hundreds of minor salivary glands, so smaller glands that line the inside surface of your mouth. And often these are the ones that we sample when we do a biopsy to try to diagnose Sjögren's in children or in adults. And so when we look at one of these minor salivary glands under a microscope, you can see here on the left, this is a normal structure. And so there are a variety of parts of the gland that produce all the proteins and the liquid in saliva, and then they flow through those sort of bigger circular structures you can see at the top, which are the ducts. And when someone has Sjögren's, the immune system invades these glands and attacks the different structures. And so on the right, you can see those little purple dots. Those are immune cells that are in aggregate, so they're clustering in there. And we call this focal inflammation because there are distinct clusters, and you can see a couple of them, and I got a little crazy with arrows here, but there's one kind of on the left, and then there's another cluster on the right, and we're able to quantify how much of this focal inflammation is there to help diagnose Sjögren's. And so what my lab is interested in is understanding why these immune cells go into the gland. How are they getting in there? What are the signals that are telling the immune cells to do what they're doing? But we know that these immune cells begin attacking the glands many years before the diagnosis is made, which makes it very, very difficult to study the beginning of this disease in people. And because of that, we turned to a mouse model, which actually develops spontaneous inflammation of these glands that's very similar to what we see in people. And so this is a slightly zoomed out view of the glands, but you can see on the left is a salivary gland with a lot of normal tissue, but then those clusters of immune cells that are not supposed to be there like that. And on the right, you can see a similar picture of the lacrimal glands. And again, it is these clusters of immune cells that we want to understand why are they in there and what are they doing, because this looks very similar to what we see in people. So the hope is that this will then give us insight to the beginning stages of this inflammation in Sjögren's. And so now we have a model to be able to study this, but to understand how these immune cells are communicating, well, we have to take a step back and understand how immune cells communicate. And so there are multiple ways that this can happen. But one of the ways I'll mention, because I think it will be relevant for the next talk, is that some immune cells really kind of communicate in close proximity. So they each put out a protein on their surface, and those proteins will essentially connect, or they'll sort of approximate right next to each other, and that will set off a signal to both of these immune cells telling them something important. But the signals that I study are actually the secreted proteins. So this is another way cells can communicate with each other. One cell can secrete a protein, and then if the other cell has a receptor on its surface that can detect that protein, then the cell with the receptor then will get the message, and that will help the cell to know what it should do next. And so there are many different proteins that can be used by immune cells to communicate in this way. But one pair that I'd like to talk about is interleukin 27, or IL-27, and the IL-27 receptor. And so this is a protein and a receptor pair that we were wondering, amongst many, are these two, are IL-27 and the IL-27 receptor, required for these Sjögren's mice to develop the spontaneous inflammation that they get? And so in order to study this, I guess first I will tell you that we know that, and this is an even further zoomed out picture of one of the tear-producing glands, where you can see lots of inflammation in these Sjögren's mice. And we know that there are cells in the glands that are making IL-27, and we know that there are immune cells in the glands that express this receptor on their surface. And so to be able to understand how important this is, we actually began to study mice that were genetically modified so that they either could not make the IL-27, but they still had the receptor, or mice that could make IL-27, but they did not have the receptor, so there were no immune cells that could detect it. And it turns out when we look in the glands of these mice, we see that they don't have inflammation. So there's no gland inflammation in the mice that cannot make IL-27 or in the mice that don't have the receptor. And this told us that this is a pretty important protein receptor pair for the development of this Sjögren's-like inflammation in these mice. And we're actually doing a lot more studies to try to really define, why is this signal in there? Where is it coming from? And what is the signal doing to the cells with the receptor? How is it telling them to do something that really we don't want them to be doing? But I'm not going to get into that now, because I guess you guys don't have all afternoon. But I will tell you about another study that we've done that we're ongoing as well, where we're studying to identify proteins in the saliva of children with Sjögren's. And for this, we have collected some saliva from children, so 11 children with Sjögren's at the time that we did this initial study, and we had 16 children that we collected saliva from who did not have Sjögren's or did not have any other autoimmune disease diagnosis at the time. And we were able to measure proteins in their saliva. We actually measured over 100 proteins, but the one that was the most striking was, very excitingly, IL-27, which was very obviously different between the two groups. So when we measured it, that's what the graph shows, there's more IL-27 present in the saliva from children with Sjögren's disease compared to the children who did not have Sjögren's. And so we think that this suggests that this is a relevant protein that is playing a role in the autoimmunity in children with Sjögren's, which we believe is a relatively early stage of disease. We still need to do more work, but it's promising in my opinion. And so really to kind of summarize what I've told you, Sjögren's disease is a systemic autoimmune disease that is difficult to study early on in the disease process, but we have mice that spontaneously develop similar immune cell attack on the saliva and tear-producing glands that mimics Sjögren's disease that we see in people. And we know from these mice that this IL-27 receptor pair are required for this spontaneous gland inflammation to occur, and this protein is elevated in the saliva of children with Sjögren's. And so we're hoping with this information, we can really develop tools for earlier diagnosis and responses to treatment, maybe even develop new treatments. But we have a lot more work to do, and I have a whole team of people I work with. My lab at the top, Yiguang Chen is the guy who made all the fancy mice, and then the saliva study team at the College of Dentistry, and we appreciate a variety of sources of funding to fund our work, and I will gladly take questions later.
Thank you so much, Dr. Lieberman. We really appreciate your breakdown and making that so accessible for us to understand. Now I'm really happy to introduce Dr. Dezfulian. So Dr. Mohammad Dezfulian is an immunology researcher passionate about uncovering how the immune system works and developing new tools to better understand it. His lab creates innovative technologies to study how immune cells behave in different diseases, including autoimmune disorders, cancers, and infections. By revealing these fundamental mechanisms, his work aims to pave the way for more precise and effective therapies for patients. All right, Dr. Dezfulian, please take it away.
Thank you so much, Kaylin. Thank you so much to the organizers for inviting me here today. I've had a really wonderful time listening to everyone speak, and I've learned so much so far. I'll start with a little bit of a background about myself and why I am studying Sjögren's. So right now, I basically set up my lab at Penn. We set it up in January, so relatively a new lab. But I kind of grew up in Iran, and I've been living all over. I've lived when I was a kid, I moved to the UK, then moved back to Canada. And then from Canada, I moved to the States. And then from here, did my postdoc, and now I'm in Philly. So I've kind of been going around a lot. But basically, my training and my background has always been in molecular biology. Immunology was something that was kind of relatively new to me. When I was doing my PhD work, I kind of realized that in the field of biology in general, specifically in immunology, there's definitely this need to kind of develop new technologies, because the current technologies that are out there are not really allowing us to systematically and unbiasedly answer some of these fundamental questions. So after I finished my PhD, I joined Steve Elledge's group at Harvard Medical School. And over there, we decided to answer one of the most fundamental questions in immunology. And that was, what do immune cells, specifically the T cells that Scott really beautifully laid out earlier, recognize in disease states? And the way we did that was basically, our lab decided to address the fundamental question of how do these T cells recognize their pathogens or their antigens on their target cells? Now you can kind of think about the immune system as a whole. There's a lot of different cell types within. But one of these really important cell types are the T cells. Now, the T cells are, imagine the like keys, they can recognize specific antigens. And the antigens, you can kind of think about them as doors. Now, in your body, each one of us has kind of millions of different T cells or millions of different keys. Each one of these keys opens a unique door or antigenic fragment. Now, typically in a normal setting, these cells have evolved to recognize only pathogens. But in a disease setting, for example, in the context of autoimmune diseases, these T cells start attacking these healthy tissues using their keys. The keys that have evolved to recognize pathogen are kind of now redirected, and they start attacking self-antigens that are being processed and presented. So, and the reason it's been difficult to associate these T cells or TCRs with their cognate antigen is because of the high level of diversity that you have. Every individual has millions of different T cells. You have multiple different HLA alleles across the populations. You have thousands of different HLA, each one of these doors, and each one of them is now presenting a unique antigenic fragment. So the question of kind of combining what these T cells recognize with their specific antigen has been a question that has been kind of, a big challenge in the field for the longest time. So the focus of my research has basically been trying to develop tools and technologies that would allow us to bridge this gap. And the way we've done this is basically we've developed two technologies, which I don't want to go into too much detail. Now, one is for a subset of T cells in the body called CD8 T cells. The CD8 T cells are cytotoxic, meaning when they attack a tissue, they start destroying it because they have a set of enzymes inside of them. And on the other hand, you have the CD4 cells, which are more considered to be helper T cells. They don't directly kill your target cells, but both of these are the two major subgroups of T cells that you have in your body. And we developed these platforms and we published them in 2019 and 2023. Basically, both of these platforms are the first set of technologies that are allowing us to systematically decipher what these T cells recognize in a really unbiased manner. Once we had the technologies, one of the key components of us when we were trying to study autoimmunity, in the context of autoimmune disease, typically what happens is that in your body, you have thousands of different proteins. So if you look at the human genome, we roughly have 22,000 or so proteins. Now, each one of these proteins could potentially be recognized in an autoimmune disease. So in order for us to develop the platforms, to develop these technologies, we need to assess each one of those, because in most of these autoimmune diseases, we don't necessarily know what specific protein gets recognized and targeted by the immune system. In other words, that T cell, the key, we don't know which one of these antigens, which one of these 22,000 or so proteins is being recognized by that T cell. So what we did is we decided to synthesize everything that we can find in the human proteome. So we made a big library of antigens, and then we expressed them in our target cells. Now, these target cells, once you have every single protein, then we basically can take T cells, either from a healthy individual or from a disease in the context of Sjögren's, for example, and then we can take those and systematically try to identify which specific one of these fragments the T cell is recognizing. Now, we've done this extensively across different contexts and pathologies, but something that was kind of we decided to do early on was we focused on Sjögren's disease. I'll be honest, when I started working on it, I basically made a list of different autoimmune diseases, and I realized in Sjögren's, it's relative to other autoimmune diseases, it's kind of neglected. We don't know anything about the antigens that these T cells are recognizing, despite the fact that we know these T cells are important. So now Scott mentioned, one of the main hallmarks of the disease is the fact that within the salivary glands of patients or the thyroid glands of these patients, you have these different cell types. For example, every tissue has, in the context of salivary gland, you have different, like you have acinar cells, ductal cells, and within those, the T cells come in and they start attacking these tissues. Now we know these T cells are clonally expanded. Clonally expanded basically means that there's more than one of the same thing. Now that is an indication that most likely these T cells are recognizing some kind of a self-antigen or a self-protein within the tissue. And we think, based on whatever's kind of been published in the literature, that potentially the ductal cells, so this is kind of one of the subsets in the salivary gland, is the one that's being targeted. So the question that we had was, can we identify self-antigens in patients that these T cells are recognizing? If we know these antigens, we can not only understand the disease better, but if you really think about it, if we know what it's being recognized, you could potentially block that recognition in a more of a specific manner. So we took these T cells, and this was our experimental design. The way it works is basically we collaborated with the Dezfulian group at the ANRF, and we extracted memory CD4 cells from patients. So these are CD4 cells that are clonally expanded, and we think they've seen an antigen. And we took these CD4 cells and we plugged them into our T-scan platform, specifically the T-scan2 platform here, because that's the one that recognizes the antigens for CD4 cells. Once we had these T cells into our platform, we can now systematically try to identify what they're recognizing. And sure enough, for a subset of these T cells, we were able to identify self-antigens. For example, this is one of the antigens that one of these T cells are recognizing. So for the first time, we're able to identify what the specific antigens or what the specific fragments are that these T cells recognizing. Now, once you know these antigens, you can go out and start asking more fundamental questions. For example, now that we know what the specific protein is that it's being recognized, the first question that I wanted to answer is that, can I use the platform that, the antigens that we've discovered to figure out which specific cell type within the salivary gland is being targeted? Because as I mentioned, there's multiple different versions of these cell types that are identified or found within the salivary glands. And we have this kind of really elegant technology where it's called single cell sequencing. So you can basically decipher all the different cell types within a tissue. Now, what you're seeing over here, unfortunately, it's a little bit blurry. Apologies for that. But the darker cells, each one of these colors is a different cell type. So the darker cells are the ductal cells in the patient. So these are basically the ones that are we know that T cells kind of congregate around them. Surprisingly, if you look at in healthy individuals, you don't really have any HLA molecules. So those are the antigen presenting molecules on their cell surface. But if you now go into Sjögren patients, you start seeing class 2 or HLA molecules on these cells, which validates a lot of what we already knew about the disease. But what was really interesting is that the antigens that we had identified, despite the fact that we had done it in a really unbiased manner, you could see these antigens, these fragments, are uniquely being expressed within the salivary gland of these patients, especially, for example, this DNA damage protein. So we think that these T cells are able to recognize self-antigens that are mostly being processed and presented by the ductal cells within the salivary gland. There are other cell types there. We can't really systematically exclude those, for example, antigen-presenting cells, dendritic cells, etc. But we think, at least based on what we've seen, is that the ductal cells are the primary targets of the self-reactive CD4 T cells within these patients. Now, this is where the story gets a little bit interesting and problematic, is that once we knew the antigens, typically you would assume all the antigens would be shared across these patients, and they would have restricted HLA presentation, meaning that, when you do risk assessment, like a genetic risk assessment, if you look at the genome of across multiple cohorts of patients in Sjögren and lupus patients, typically the strongest gene that's associated with the disease is this HLA allele called HLA-DRB3. It's not really important what the allele is, but what was interesting is that the loci that's associated with the disease is a specific HLA allele. Now, the antigens we identified didn't seem to be all restricted on HLA-DR. So the question well, if the T cells are recognizing self-antigen, and this is the strongest risk factor, why aren't all the T cells recognizing antigens on this specific HLA allele? So we went back and we looked at the genetics of the disease and how it was done. And we realized quickly is that when you look at the GWAS analysis, these kind of studies that are done to identify the risk alleles, is that adjacent to the HLA alleles, when they did the initial kind of GWAS analysis, these studies, they were mostly done in the European population. Now, within the European population, adjacent to the HLA allele, right next to it on the chromosome, there's a set of other proteins that are called complement. They're again part of the immune system, but they do a function totally different from the HLA alleles. Now, because they're kind of really associated or they're kind of close proximity to each other, everyone that has the DRB3 allele typically has a mutant form of the C4 allele. So it's like you can have both of these at the same time. So that kind of led us down this analysis. Is like, well, how can we actually decipher or figure out what is it that's actually driving the disease? Is it the HLA allele? Or could it be that the complement, the mutant complement, is playing a major role in the initiation of the disease? So now, if you expand the GWAS analysis, and this is what Steve McCarroll's group did in Boston, is that they expanded it and they included more African American people with African American heritage in the studies, and they realized most of the phenotypes that we see in the patients are always now associated with the C4 and no longer with the HLA-DR. So it's basically a genetic way of kind of dissecting out which one of these is the main driving factor for the disease. And we think, based on what we've identified, that it's mostly the complement. And that actually fits nicely within our model too, because if complement is the major thing that's driving the disease, all the proteins, all the antigens should be across multiple different HLA alleles. In other words, because the main function of the complement protein is to clear cell debris, pathogenic fragments, if you have lower levels of complement, you start having accumulation of antigenic fragments or these proteins within your tissue. And all of these could ultimately be presented on multiple different HLA alleles. That's exactly what we were seeing. So basically, our studies showed us or told us, because we were doing them in an unbiased manner, that most likely what's initiating the disease in a subset of patients, now I don't know about all patients, but at least in a subset of patients, is the lack of complement and not necessarily HLA alleles. Now, something else that's also mentioned, and I know someone had asked in the in the Q&A at the bottom too, is that could the microbiome also play a role in the development of the disease? And that's something that we can also address using our platform as well. Because, once we know what some of these antigens or some of these proteins are, we can then go back and do experiments using our T-scan platform to figure out if there are any bacterial antigens that are similar to our self-antigen, for example, the MAP kinase that we had identified. And sure enough, for a subset of our T cells, we were able to identify bacterial mimics of the same antigen. In other words, if you really think about it, it's like if the key is the T cell, you basically have a key that can open two doors. One is the self door, is the self protein, but that same key is opening up a bacterial door as well. So both of these are being recognized simultaneously using one key. This is called cross-reactivity. Basically, it's a key that's doing both these things. And we think that potentially these cross-reactivities could be playing a role in the development of autoimmune diseases. Especially like now, if we go back and we looked at the bacteria that we can identify in the oral microbiome of patients, the abundance of the bacteria that we identified as being the target for some of these T cells is higher in the oral microbiome of patients. So we think this cross-reactivity could also play a role in the development of the disease. Now, to summarize, basically what we know is that, we've developed a set of technologies that are now allowing us to systematically address some of these basic questions in the field. We can now take any T cells from autoimmune patients and kind of unbiasedly understand what they're recognizing. We think, kind of in the context of Sjögren's, these T cells are recognizing self-antigens. Now, these self-antigens are potentially being expressed on multiple different HLA alleles. So that means every patient is going to have a unique set of antigenic fragments that are all contributing to the disease. Using the antigens, we identified that most likely it's within the salivary gland, there's a specific cell type, and that is the ductal cells that are being targeted at least by the CD4 cells. And again, we think that complement plays a major role in the development of the disease. Now, I want to kind of highlight here that we're not saying that the T cells are not important, but we think they're actually really important. But in the absence of that initial trigger using complement, like in the absence of complement, it's only when you have complement or the lack of complement that the T cells come in. So it's this interplay of both of these that's essential for the development of the disease. There's so many people kind of I need to thank, but I'll kind of, my postdoctoral lab at Harvard, Steve Ellich, my mentor, it was kind of a really wonderful mentor to work with. And my lab right now, so I have two really talented postdocs. And both of these postdocs are kind of working, trying to address some of the questions that kind of come up from these studies that we've done in Sjögren patients. And I definitely like to thank the kind of funding agencies, the Sjögren Foundation, the Colton Center at Penn. One of the main reasons I actually joined Penn was because Penn had decided to set up a foundation, a center just dedicated to study autoimmunity. Anyways, thank you so much. I'll be happy to answer any questions.
Thank you so much, Dr. Dezfulian. We really appreciate you sharing all of these really incredible insights and innovations and learnings. But before we open up for Q&A, we'd love to hear from you. We would like to, we know Sjögren's disease impacts people in so many different ways, and we'd like to know what's the number one thing you'd like to better understand. You'll see the quick poll pop up on your screen with a few options, so go ahead and choose one that resonates most with you. Is it symptoms and flares, diagnosis and testing, research in the future? Wow, it looks like treatments and medications coming in, but lots of interest in all areas. So now we'd kind of like to open it up to our Q&A session. And I'd also like to mention that if you've been putting any of your questions in the chat box, in the Q&A chat box, lots of them have been answered in the actual box itself, and you can read through those answers as well. So we'll get to as many as we can. So this is a question to all of our speakers, actually, which is, are there any symptoms people should be mindful of when considering other autoimmune diseases that have been linked to Sjögren's?
I'm happy to jump in and start with this one, but maybe Abby's experience will also be helpful. But yeah, I mean, I think of Sjögren's as a condition that's often associated with other autoimmune diseases, but also can itself cause symptoms affecting any part of the body. And so I think the really important thing is make sure you have a good rheumatologist or whoever your general, primary care doctor is who can help you navigate the system. So if you have symptoms, definitely let your doctor know so they can help decide which testing needs to be done. For example, celiac disease, thyroid autoimmunity, those are two of the more common conditions that can also co-occur in people with Sjögren's. Your rheumatologist should be monitoring for things like arthritis, and they can help figure out, is it arthritis related to Sjögren's, or are you developing rheumatoid arthritis? And then there are other conditions as well. So that's kind of a probably unhelpful answer.
I thought it was helpful. Yeah, I completely agree. I think the point that it's systemic and it can be progressive complicates this a lot. And especially as Dr. Lieberman's saying, we know it can probably be showing up in peds, right? So this presentation can change over time. So I think as a patient, being aware of new onset changes and what the top maybe 3 symptoms you're dealing with at any time, because your appointment with your doctor is often 15 minutes. So if you come in with a list of here are the things that are affecting my quality of life, and my ability to act independently, those are the things they're going to act on first. So having a list of those to me has been the most helpful. If you are having, issues with brain fog and issues with pain, that sometimes can be, oh, I think I'm aging, or maybe I'm starting perimenopause, or, there's other confounding things that can be going on. So, advocating for yourself, bringing up those facts to your doctor. And then if it is a neuro-related thing, that often can be a little bit of a hot potato, with the rooms not really wanting to own it, the neuro not really wanting to own it. And I think that generally speaking, like neuro Sjogren's kind of ends up being neuro as the lead instead of rheumatology. So, maybe making an appointment with a neurologist if you haven't done that yet, ask for a referral from your room. That might be something helpful if you're noticing issues with pain, or if you've like previously been diagnosed with fibro, things like that.
Great. Thank you so much. So kind of another question to everyone, and Mo, if you want to take this one first, but what are the latest discoveries in the field that really excite you?
I guess that's a really good question. So, a couple of different things is like I think Scott mentioned this as well. Like we have kind of a really major challenge in terms of kind of studying the early events. When a patient comes through the door, it's usually years after the onset of the development of autoimmunity. So the question is, how do you study those early events? And I think we need technologies or tools to better address those. Now, Scott is doing a really elegant work using mouse systems. Something else that's kind of Dr. Blake Warner and I have been discussing extensively. Dr. Blake Warner, he's a Sjögren researcher at NIH, is that potentially we can study these in the context of other diseases. Now, cancer patients, when they develop, they undergo immunotherapy, basically what the immunotherapy is doing is unleashing the immune system. It's just you're giving them this immunotherapy drug, so that's unleashing the immune system. Some of those patients go on and develop, autoimmune-like features. So, for example, you have Sjögren-like features, diabetes-like features, etc. Because you have this before and after, before the administration of the drug and after the administration of the drug, we think those could be kind of a really unique window to study those early events. And so that's something that we're kind of extensively going in as well. So a combination of like being able to study those early events with our technologies and some of these spatial and technologies that are coming out, it's really allowing us to kind of go after those early events, which I think is really important if you're trying to study autoimmunity in general.
Can I jump in just one quick point? I just want to say that I am very excited to have my co-speakers here involved because we're getting new technologies. We have people studying the nervous system and the effects. And so I think the fact that people are now starting to be interested in studying Sjögren's from all these different angles, I think is really where it's going. And so I think that's wonderful. Thank you, guys.
Thank you.
Thank you.
Well, thank you. Thank you all so much. We truly appreciate it. Unfortunately, we have to wrap up and we have lots of questions. And again, if you didn't get your question answered, make sure to email us at info@curearthritis.org. So as we wrap up again, I just want to say a huge thank you to our fantastic researchers and speakers for sharing your innovative work towards understanding Sjögren's disease. You can see firsthand the impact ANRF and Sjögren's Foundation have in supporting these incredible individuals in their quest for a cure. So thank you to everyone who joined us for this great event. If you've enjoyed the webinar, we kindly ask you to consider making a donation today by visiting curearthritis.org/donate. Every contribution helps, and it helps us bring these educational series and research around Sjögren's disease. We'll be sending our live attendees a survey after the event. Your feedback helps us find direction for our future webinars, and please share your comments with us so we can continue to provide important and relevant education and content to you. And please don't forget to sign up for our other Research Spotlight series. We'll be live at the same time on October 22nd with rheumatoid arthritis. Thank you all for coming. And please remember to follow us on social media as well, just to stay in touch and connected with ANRF and the Sjögren's Foundation.